Ashmeadiella, I'm going to run the Genera scripts thing here in a second, but I just wanted to show you what it looks like. In the east, this is relatively uncommon to very uncommon species. As you move to the prairies and as you move to the west, it becomes more and more common. You know, it's not the kind of thing you run into every day, but it's reasonably common, and I believe is a hole nester in wood or stems. So the vibe and the size you can see from the pin here. This is a female. Well, no, it might be a male.
Ashmeadiella?
This is Ashmeadiella, and this is Ashmeadiella floridana. So is very much like Hyriades, and we'll see a couple very clear differences. But if you looked at it very quickly, you would say you had a Hyriades, which is a much more common species. And in this case, let's flip to the B genera memorization chart for a second and catch up mentally with all this running around with our heads cut off. The power, something bad was happening to the power at the whole research center, and they had to shut it down. As we approached the house, it was like, internet's back up, but we are dedicated and came in. Where is, there we go, Ashmeadiella. So we look at Ashmeadiella, I want to say that is going to be a male over here. And we go down. So like the group as a whole, which almost all have, let me pull this down just a little bit, has 3 submarginal cells. I mean, 2, the group as a whole has 2. Let's see, just going to make sure I am in the right spot here. That was, I changed to Osmia, and I want to be in Ashmeadiella land, going over and back. Okay, so the male, so there are no pale markings. So Stelis, which some of the Stelis subgenera look a lot like this too, have pale markings on the abdomen. They have none. The aurolium is present, as it is in most B genera and species. If the aurolium was absent, you would lean towards Dianthidium. I don't know if there are other megachiles besides megachile or megachilid family members that don't have auroliums other than megachile. Maybe we'll surprise ourselves. And you'll see what we're talking about here, the anterior surface of the mesepisternum. So that's the side, the plate on the side of the B, and it's the surface that is to the front, so that would be facing forward. And what we're talking about is that rather than that being a simple curved over component, the front side and the lateral or the sides, there's a sharp line. So you'll see it's almost at a right angle, and then there's going to be a raised line, or a carina, we would call it, between those 2 sides. And we'll also see that, unlike Hyriades, you don't have the open cells along the propodium. That's not necessarily in here, but it is also good characters, or the raised line going around T1. And additionally, we're going to look at the rear end and see that there's 4 projections, or they would call them teeth, on T6. So that's the last part of the abdomen along its, or the visible part of the abdomen along its rim. So let's see if we can find those things. And if we flip back here, so here again is Ashmeadiella, and I don't have, I can use my thing. So this is the mesepisternum here. So orientation, there's the tegula, the front wing. So the big plate below the front wing is the mesepisternum. Let's see if I can shift this a little bit there, and a little bit more in focus. And then normally this side of the plate and this side, which is the front side, would simply curve over without any sharp demarcation between the 2 faces. it would just, they would merge to one another. But here, and we'll zoom in a little bit, there's a very sharp line. This line here is abrupt, and there should be a carina in there. Let's zoom in and see if that is visible. I have a female that also is true also, and we should be able to see that. So there's a lot of hair here, so and I'm not seeing, let me get the white balance going, and the it's not, so I think the important thing is that this face is the anterior face, and this is the side, the lateral face. This, although hard to see here, maybe I can go all the way up in terms of power. Yeah, it doesn't really resolve well, but there are, there's a raised line along here. You're seeing all this hair on the lateral face and no hair on the front face. This is often the case. Part of what's going on is these legs can tuck up into there. And so this bare area is partially a slot for the legs if they have to crawl into a hole or otherwise become transformed to a small tubular thing. So that's part of it. We'll revisit that in another specimen. Let's look at the propodium area. Although this particular specimen, I think it is cocked up a lot. So it may not resolve really well here, but I think you can see in the metanotum and propodial area, if this were a Hyriades, let's see if we can get the white to be brighter. Apparently not. That you would see a whole series of large open cells. And in this species, like a lot of others in that group, like Megachile and Osmia, it's just, you know, it's there, but just as a smooth transitional line. Again, we've got some very dark specimen on a very light piece of paper, and I don't know if I can change that with my finger here at all. Yeah, it's still difficult to see. Anyway, you don't see these big blocky open pits, and then we can't see the T1, the front, the anterior face of T1, because this abdomen is cocked up. Oh, this is a female. Never mind, I was wrong about that, but the characters hold for both at this level. So you can't see that there is no carina there separating the two faces, which Hyriades has, and is sort of the giveaway for Hyriades. So let's go to the male and take a look at it. So those are the primary characters for the working with Ashmeadiella in the females. It's that character of being a small dark bee in Megachilidae group with the two submarginals and with hair under the abdomen, and then the presence of that very sharp line between the anterior and posterior parts of the lateral plate, which we will call the is the mesepisternum. Sometimes in the old literature it would be the pleura, and I'm sure there's other names too. So this is not formed well, so I'm going to change the position, but there would be the anterior. There's something of the sharp line is showing up there, but I think perhaps on the other side of the specimen we have a better view of the carina and the two faces. So as dark bees go, you know, again, we run into these problems where seeing detail within the dark areas is difficult. So there, the front leg is tucked up into this area. Again, just not getting enough light in there. Oh, that brightened it up some. So carina is here. This is bare, but it's this face here and this face here. Very sharp line, I guess, is the point. I'll try one more time on the other side with this extra light that the microscope has given us to see if we can't pull out that line at all. You can't if there's a wing in the way. Yeah, I'm afraid
You want to redo the
less
white
than.
balance?
Right now, it's more that the wing is hiding, is in front of the area that would want to see. Anyway, so this is a male. It does have that same feature. Let me try from the top one last time on that side. So what you're looking at here is a tegula, scutum, wing going off. This is the plate, which is the mesopisternum or pleura below the front wing. And here is the sharp line. There's the anterior face. Its legs are going to tuck into there. And here's the lateral face. And there's a sharp line, which you can kind of see here between the lateral, more hairy, and the clear and glossy interior space. One last time, we'll try and zoom in there and see if the raised line part is very visible. But I think you are getting the point of what you should be looking for and would not be there if it were a Hyrides. So somewhat better. Never can tell what you guys are seeing here. Here's the two plates, or the two sides of the same plate. This is the lateral side. Here's the anterior. This is the sharp line, and it is pretty visible. This is as high power as I can get on there. But if you're imaginative, I believe you can see that there is a raised line. You certainly can see that there is a sharp transition between these two faces. Would not be there.
Can you move the cursor out of the way so I can screenshot?
Yeah.
Great.
Would not be there otherwise.
Thank you.
All right, and now I believe we can go to see the abdominal area, which is bent over. This is always the plus of working with specimens that are not put together by a carding. So here, this has the opposite. So in Hyrides, there was a raised line that went all the way around here, and you had a relatively sharp transition from the top and the anterior face. You still have an anterior face and a dorsal or top face, but you don't have a sharp line here. It's a transition. It just smoothly moves from one face to the other. I'll zoom in here a little bit. And got a lot of magnification. There does seem to be some kind of line there, but it's not at that same level. This is not a good shot here. This is Ashmeadiella opuntia. Let's see if I can bring it around this side. And also the raised lines, or the bit large open cells on the other side are not present either. So that's not the greatest look there.
Yeah, we can see it.
Okay.
Just bring it up a little bit higher on the screen.
All right, it will shift to the rear end. And in the males, as was pointed out there before, there are roughly, there's some kind of teeth at the rear end of the.
So we're looking at a male right now.
Yeah, we're looking at a male. And Ashmeadiella opuntia. You can probably tell what it specializes on. And there are two lateral ones here. Let's see if we can change the white balance. doesn't seem to want to. Let me see if I can shift it a little bit so you can see through the hairs. There was actually, that was pretty good for a second. This might be a slightly better view here. What we're looking for, yeah. So here are the projections. There's two on the far sides. These two central ones are more visible right now. Let's see if the trick of putting my finger below the specimen is helpful. A little bit. There we go. Oops, now you can see the teeth through the hairs a little bit better. So they have 4 in the back. So that's the relatively uncommon Ashmeadiella, at least in the east. You're only going to see that in the south. And I've only collected a handful. But these others, like this Ashmeadiella opuntia, was something we picked up in the Badlands. So as you move west, you'll see more of those. All right, let's go to Chelostoma, which is also in that group of genera that are small and black and megachilidae in
what do we have in front of us?
relates. This is Chelostoma philadelphia. We'll jump over to the
Wait, we have some Ashmeadiella questions.
Okay.
1 second. Man, I think slower with connection. Are the teeth on the male a genus level character or a species level?
So it could be both. I don't know enough about telling the species apart, but my notes in the genera guide indicate that it's a genus level. But I have relatively little direct experience with the species other than just literally 3 or 4 that I've ever collected. So someone could speak up, but I think that the 4 teeth are a genus level ID character because I put it in the genus memorization chart. So, but I don't know where I got that. I could have gotten it from Michener or another group too. So we're looking at Chelostoma now, and the thing to note, this is, I believe, a male, and the thing to note about it is it's really, really skinny, and that, we'll go back to the memorization thing, is, and the general lack of characters that it has. Otherwise, it comes down to basically being a skinny black bee in the megachilidae group. So we're looking at a male, so let's go to the males here. So it has the typical submarginal cells and has an aurolium, no pale markings, so that would be similar to Ashmeadiella and Hyriades. And here it just basically defines slender as the shortest distance between the tegula, so those are the 2 wing covers, less than or equal to the length of the scutum. So you're looking on the scutum, and the scutum dimensions are longer along the axis of the body than wide. And so let's see if we can visualize that here. And that's basically the ID string. Now that we're here, let's look at the female characters. And yeah, it's exactly the same. So if we go back now, let's see if we can note how skinny it is. So it doesn't have like the others do, which is a very noticeable set of characters. It is skinny, dark megachilidae. So here is, well, let's zoom in. Here is the scutum and scutellum area and the bee, and I'm going to flatten this a little bit more so it is as right angles as possible with the camera angle. Okay, that's pretty good. So so this particular species is our native. There are two non-native species in the region. Those are in the northern part of the east. I'm not sure if how far south they will spread. So here's the tegula. This is the scutum right here. So this distance here is shorter than this distance from here to here, the length. It looks almost equal, but I think I would agree with that statement that this distance here is shorter than that distance. That's what you mainly have in terms of, if you go through, yeah.
Before you move it, will you give us a zoom in so I can screenshot?
Okay.
And I'll do a little diagram on that one when I do some photo editing soon.
So our native species is what people would say in the literature is a Philadelphus or mock orange specialist. But the place you collect it the most is on the old-fashioned mock orange that your grandmother, for example, would plant in their yard. Most people don't plant that tree anymore.
Move the cursor.
But it's a bush, but it's actually not a native Philadelphus. It's a introduced one, and I don't know. So the native Philadelphus is that group is very uncommon, and there's a question as to whether it really is a straight Philadelphus specialist in the wild, because we have found the species, and Mike Arduzer has too, in locations that have no known populations of mock orange present or not. We came up with a theory as to what it was using, but it now escapes me. It might be that it's using black gum, which would be interesting.
Nessa just showed up in the chat.
Okay, so that should be checked out. There's records from Suzanne Batra's work, which is a very tiny study on black gum that should be investigated. So, and you can see, there's other features here that it does not have that are going to be helpful. Like it doesn't have the carina separating the anterior and the lateral faces of the mesepisternum, which would be below the tegula over here. It does not have, as you can see back here, get it in focus. It does not have a carina around this face, so that eliminates Hyriades. Right here, there are no pits; it's more striations. So that fits in, or maybe maybe you could call those pits. So I'm going to take that as a feature.
Yeah. When I first learning, I mistook it for Hyriades because of those for a while.
Yeah. So anyway, the basic thing is long and skinny, and the specimens, the introduced specimens, one is a specialist on campanuli, and both the introduced campanuli that's all over the place up north, and probably our native ones too. I don't know. But
what did you mean by the Philadelphus species?
Philadelphus is the genus of mock orange.
Okay.
And so almost all the collections of this species come from collecting off of mock orange because it's in everyone's yard. It's very available. It's being used a lot by this species, and things like black gum are uncommonly collected on because it's a tree. And additionally, we just find them in, kind of randomly in bull traps and other places that we can't quite put our finger on as to why it would be there. Or on something like blackberry, which is attracting a large supply of other species. Oh, is that the male? Yeah, so let me just point out that the rear ends of this group are also, of the males, also have lots of architecture that are useful at the species level. So the two introduced and the native one. Gosh, I'm not even sure if there is another Philadelphus in the West or in North America at all. This may be the only native species, for all I know. But if you're identifying to species, you're going to be looking at these, if I can get this done, these teeth back here. So this one possibly may have 4, which would match that character in Ashmeadiella in the male, but I believe the others have, you know, things that are modified into plates and scutes and that kind of thing. It's illustrated in the Discover Life guide, but you end up looking at the rear end a lot.
I just want to read out loud a comment about the mock orange Nissa sylvatica thing.
Okay.
So Heloise writes, order is Cornalis. Families include Cornaceae, Hydrangeaceae, Nissaceae, and more. So those are all relatives to what he was just talking about.
Okay. Yeah, so all of that I think needs to be investigated, because I don't think that the explanation that it was a specialist on mock orange is sufficient, because the native mock oranges are just really uncommon. Could be though still, but we think there's more going on. And so yes, please look. This has a pin right through the middle of this female, so it's not. Let's see if we can get some light on this. Not very well, but again, it's the ratio between the tegula and the length is the important part, but it is not illustrating here well in terms. I'm trying to fade the light with my hands in this shot, because this is my only other Chelostoma specimen, so I have no more to show, sadly. We give away too many things. Okay, now we're going to jump to the, I believe there. Pardon? Oh yeah, if you put that on. So we're going to jump to the Dianthidium groups. So colorful, lots of yellow markings, definitely not all black. And we're going to do Dianthidium first. And the vibe here, let's get that in the picture, is definitely, you'd look at that and go like, well, I could be looking at an Anthidium. What
do you have up there?
This is Dianthidium curvatum. No, consinum. And we will get to its characters, but let's read them out.
Are all Chelostoma potential specialist species? We have 2 in the UK that are both specialists, bellflowers and buttercups, says Ellen.
You know, I think we have both those over here. We have Chelostoma relunculus, I think it is the name, so that would make sense from the buttercup, and the other is Chelostoma, let's see, Philadelphia is a native, ranunculus or ranunculi, and then I can't recall the last. So I think, so I can't answer it completely, but I think we have all of your specimens, and we certainly have introduced and native buttercups out the wazoo. Okay, so let's go to Dianthidium here while people chat, and let's get out of this, and we'll go up.
So just for those in the chat, I know this doesn't help with the recording, but I'm copy and pasting the names directly from Discover Life, so where I have it in the chat is actually the link to the species page, if that is helpful to you.
All right, Dianthidium. So there's a couple things here. One, in the east, they're relatively uncommon, often associated with sand. Out in the west, they're much more common. And when you're looking at them, you'll see this, that the pronotal lobe which we'll point out in a second, is very elongated, plate-like, rising above the bed of the scutum, and the edge is transparent. It has the usual 2 submarginal cells. It's got the yellow, white, or ochre markings that basically that Dianthidium group have. Unlike Dianthidium, it does have an aurolium, so that separates Dianthidium if there was a question. It does have scopa, so that means it's not a parasite in the female. And then you have just what we were talking about before. You have the anterior face at the mesopisternum is flat, and the lateral face is marked by that junction is marked by this carina or raised line. And then I believe in these specimens it's a lot clearer. And then in profile, the anterior edge of the scutum rises abruptly with above the, like a cliff, it says, above the junction with the pronotal collar. So at right bodies, we should be able to show that too. And then more in detail is the apical end of the middle tibia with one outer spine, or sometimes just a tooth. The end of the tibia is unmodified and similar to other Dianthidiines. So it doesn't have some of these things that I believe like Paranthidium has. But we'll look. I don't use that spine thing very much, because you'll see that the other characters, the 2 faces of the mesopisternum and the raised pronotal lobe and the little cliffy thing on the scutum are really nice characters. So let's go back. So here again is our wide angle view. So we're going to be looking at, here's the anterior edge, so anterior towards the head edge of the scutum, and the pronotal collar is over here, so we can't even see it, but this face we'll try and get an angle on, should drop sharply off, and not like in most of the other genera of bees. It's not just a smooth plate butting up at the same level to the pronotal collar. Over here is the pronotal ridge. It's not, you can't see it well here, but when we change the angle, you'll see it. And this is very much raised up above the surface of the scutum and has a clear, transparent edge. And this isn't illustrated well here. Here's the mesopisternum again. This is the lateral face, and there's a angle which you can't see here, but we will check out that with the anterior or front face that is very sharp. So let's first jump to see if we can see the lovely raised. And I should also point out that almost always another feature that isn't really mentioned here, but is I think useful, if I can get the white balance to be a little bit better behaved, move that over here, is that the tegula, I believe, is always D-shaped. So if we zoom in on that, let's do that here. You'll see it's large and not oval. And that maybe should be in that memorization chart. I'm not sure, but maybe it's not 100% of the time, but someone might advise on that. Let's get this in. So what we're looking at or looking for is that the tegula, so this wing cover, first of all, it's big. Oh, good. We can see the wings of the pronotal collar. So here's a tegula, very large for almost any bee, and definitely not shaped like just a simple oval. Very protective for some reason. And here is the pronotal collar edge, and or the angle, sometimes it's called. And if you look here, so normally this angle is not very obvious. It just flows with the whole horse collar shape of a pronotal collar. So the pronotal collar here is somewhat invisible because, as we'll see, this scutum is towers over it, normally you can see the very rim of the pronotal collar that connects these two angles on the side. Here we only see the angle that is above the pronotal lobe, which is over here, but this is raised up. This is transparent. I think that's pretty obvious there. There's a little bit of color there, but it's large and is standing up very wing-like in a way.
Before you move it, will you resharpen that to
the tegula?
Yeah, the tegular was better because you can still see the transparent collar that way.
All right, let's go there. How about that? And I believe the tegula often has, maybe always has, a transparent edge too. I'm not sure about that. Again, I don't see this species very often unless I'm collecting out west or looking at a Western collection. Who has time for that anymore? Okay, so let's see if we can see the edge here and its relationship to the pronotum and the cliff-like aspect, as they say. And then we'll go to the front and rear face. Dianthidiums are just full of characters. Parts of the identification. So maybe I want to twist this a little bit more this way, perhaps, to see it. Yeah, one of the issues here is you can't even see so here is the scutum, and normally, and here's a pronotal collar with the angle on it. So normally you would see a segment, at least a ribbon of this segment going all the way across and joining on the other side. But you do not, because the scutum is built up or raised above that so much that it's hidden behind it. So all you're seeing here is the edge of the scutum and not the pronotal collar at all. So if you have specimens of any other bee, you would see something. The lack of seeing something, I mean, if we were good and had a great light, we might be able to change the angle, look in here and see that segment down there. But you already have a bunch of really good characters here. Additionally, now we'll go to the other side, which I think illustrates this better. You have now a shot of the mesepisternum, so that's below the tegula. Here's the this big tegula, this big winged pronotal collar angle, and here is the sharpened face between the anterior edge, which is really not visible, and the lateral sides. So again, instead of smoothly transitioning from one face to the other, it's a very sharp edge. And if there weren't a bunch of hair there, maybe a different specimen will have this. This is actually a raised line. Here the hair seems to be right on the raised line, but when you are holding the specimen in your hands and rocking it back and forth, as is always the best way, you can see this demarcation. So those are a series of the best characters for Dianthidium, but let's see if we can flip to the other side and get even better look at the transition between the anterior and posterior side. The males are pretty much going to have the same thing. Or is this a male? Hang on for a second. What's going on down there? So it's a pollen-carrying species. Oh yeah, so there are the scopal hairs always under the abdomen in non-parasitic Megachilidae. So it is a female. The males have all kinds of cool hind end architecture that's useful at the species level. So I think here you can see the raised line a little bit better than on the other side, and a bit more of the transition between the two. Big tegula, clear on the edge, the raised angle to the pronotal collar, clear on the edge. This is the, oh, you might be able to see this quite well here. This is the cliff-like raised
right
portion.
now.
Pardon? Okay.
It's not.
Oh, I see.
Where you're...
This is the cliff-like part of the scutum, which we'll just change slightly here. And down below, way down below, is the pronotal collar that is going from here to there. And this big section here to here, that's just not present maybe in any other genera within the group and species of bees in general. So that's a good character. But so this is a rich in character genus. So again, there's a sharp line. Me's episternum is this big section here, and here's the front face glossy, and that leg is going to tuck right in there. So let's just take a quick peek at the bee, or at the male, at the bee, and then we will do a Trachusa if we have time, which I did not have time to look up, but maybe we will do some hand waving and look at it, because didn't have time because one, I did not have any specimens any longer in our synoptic collection, and two, the internet was down. So here is the male Dianthidium concinum, and just to point out here that the rear end, get this back down, is often, I think we're losing, slowing, our internet speed seems to be slow. So that at the species level, a lot of ID for the males hinges on what's going on back here. Let's see if I can change that light a little bit, maybe not. And white balance. So I'll zoom in a little bit more, maybe that'll make a difference. Yeah, it's very has wings and plates and projecting sections. I don't know if any of that reaches the level. There we go. You know, spine in the middle, two wings on the side, lots of different variations on that for the different species. Otherwise, male-wise, we go to the front of the head. Let's go from this direction. We should be able to see those same characters that we did last time. So there's, hard to see, the cliff-like face. So this is the scutum raised. We have a large D-shaped tegula and clear on the outer edge. This is the area of the pronotal angle that's raised. You can see it straight on, so it's not super visible, but you can see the rounded edge of the segment. Let's just go from the side here, and should see things a little bit different. Tegula. I'm not sure we need to spend too much more time, but the raised angle is here, and then the two faces are there. But I think we've seen those characters well in the other. So let's say we have enough time. Should we jump right to Trachusa?
Yeah, we still have 8 minutes till it's 2, but we also started late, so
right.
We have plenty of time.
All right, so I might need a little help here. I've got some really old specimens that are potentially like 100 years old, and some are saying this is Heteranthidium, which I believe is a subgenus of, oh, this says Boulder, Colorado. Interesting. A subgenus of Trachusa. And so we're going to use this as an example.
Look it up.
Oh, this is Heteranthidium occidentale, or occidentala. It's done, that's Crescen's name, so that's quite old. And there's a mosquito in here. So it could be a synonym now, for all I know. So it will look a lot like other Anthidiines.
Looks like as of Brooks and Griswold, 1988. Looks like a Trachusa subgenus.
Heteranthidium.
But you know stuff, maybe.
All right, we're going to go over here again and go and find the Trachusa thing, which I did not have time to do in advance, so we will all learn together. Again, Trachusa is really uncommon. I've just have collected a handful in the East. There is a very cool one that is a that we've been looking for but not found ourselves in Maryland, Trachusa dorsalis, which is a specialist on vining beans and peas, native ones. Okay, so theoretically, I'm looking at a female, I think, and we've got the usual two submarginal cells. The aurolium is present, which knocks out Anthidium, scopa under the abdomen, so it's a non-parasitic, and tergites with complete to only slightly broken, relatively uniform white or yellow bands. Okay, so instead of having like a lot of the other Anthidiine groups, sort of blotchy blobs of color, it should have lines across, and the anterior rim of the scutum transitioning only gradually to the rear edge of the pronotal collar. So I not the cliff. We'll look at that. Apical front ends of, oh, we didn't look at the tibial things, but in this case, it's mentioning, gotta get this out of the way here. Apical ends of front and middle tibia with only a short spine, otherwise non-modified. These spine things, I'm not sure. It sounds like that was what also Dianthidium had too, but let's take a look under the microscope at this ancient specimen, which might be like 100 years old for all I know. It's certainly dirty enough to be 100 years old. Okay, so there's our back. Let's look and see if we can see the pronotal collar visible. So here's the scutum. Here's the edge of the scutum. You can see that the tegula here is that oval or not? It wasn't in the B memorization chart, but I believe, yeah, that's pretty oval there. So very different from the D-shaped one. And this would be the pronotal collar area. And I can't see right now any particularly raised pronotal collar at the level of Dianthidium. As we explore this specimen together. So theoretically, we should see a good transition between, I might get a different specimen here too, between the, well, now that we're in the abdominal area, you can see the yellow markings are going across in basically thin lines. My vibe has always been that, like, oh, that's probably a Trachusa. But up here, we have more definitive things. That might be definitive for all I know. I mean, it is mentioned. So, gosh, this specimen is filthy. Probably was sitting out somewhere for days. All right, so we're looking for the transition between the front and the sides. I don't really see one. We're looking for the scutum diving in. This is the pronotal collar here. It's not a great look, but it seems to be basically merging. You know, the end of the scutum is touching the end of the pronotal collar here. Maybe a slope, but we don't have that abrupt cliff face thing going on. I'm going to pull another specimen and see if we get some better views here in this most ancient of collections. Now, what are we looking at? Oh, writing's eye. Oh, that's a cool one. That occurs in the Deep South. And some of these are from the Smithsonian. These are from the Smithsonian collection, and on our long-term. Okay, so abdomen lines across. Long-term goals to get these data entered, because pretty much, except for bumblebees, nothing is entered in the Smithsonian.
Male or female?
It is a female, packed with pollen. What kind, though? We will maybe.
Did you say it was writing's eye?
Writing's eye. Let's see, where does that say that's from? It's from Georgia. Vidalia. It's probably feeding on onions. Just kidding. But maybe not. So, but that is very rarely seen species these days. You have to be in good habitat. All right, let's see if we can see some of these characters a little bit better here. So we were talking about oval tegula. The pronotal collar is right through here. You can see the whole thing. There's no really raised section to the pronotal angle. The scutum seems to slope down and pretty much meet the edge of the pronotal collar. I don't see any on the side. I don't see any notion that there's a angle between the two plates. See if we're trying to get a better view of that area.
Paula shared that they have one male and one female occidentalis out of Durango, Mexico.
Okay. Well, looking at it from this direction, there might be a sharp transition. That looks, well, I can't quite tell, but it looks like it might be, it's not mentioned in the guide, but that looks like it might be relatively sharp, but it doesn't have these other features that Dianthidium has. And I'm not convinced that there is a sharp transition. Looking at it from this side, our friendly writing's eye. Let's zoom in a little bit. They're fairly big. They're, you know, compared to Anthidiums, they're bigger than almost all of them. All right, so there's a lot of hair here, so I can't quite make a declaration as to whether the lateral face and the anterior face is sharply divided or not. But I believe, based on experience, that it is not, that there's maybe it's rounded over and maybe relatively quick transition from the two faces, but I don't believe it lacks a carina in the abruptness that the other has. So I think that will do it for now. A lot of these species here, we're looking at more Heteranthidiums. Let's see if there's a male in there somewhere. Boy, these are old. Anyway, I am going off and I'll stop, but I will show some ancient specimens here and see if there's any questions. If you have questions about building buildings out of straw bales, I could also answer those.
Is your speaker on, just in case somebody opens up? We don't have any questions lingering in the chat, though. Okay. I just put in, if anybody wants to give us a suggestion for what family we move into next week. We have a few halictids left, a bunch of apidaes. Sam, do we have any, I'm going to mess up this pronunciation, Copolicana?
Copelicana.
Copelicana?
There might be some in the little Smithsonian collection that I got from Suzanne, but I don't have any.
Okay.
But we might have some.
Well, otherwise, we've already done the Calides that we have. And I don't think we've done any Melitidae. So Halictidae, Apidae, Melitidae, what do you want to see next? And if you have a question, put it in the chat or speak up. I don't think that we've done Anthophorini yet. Oh wait, did we? Did we just do that a couple weeks ago?
I think I want to say. I thought we did do Anthophora.
Yeah, we did those at the end
just
of August.
in contrast.
Mm-hmm. So if you want to go back to the recordings, I'll put the link in the chat. We did the first half of the Eucerins on August 25th and then finished up on September 1st. Could we go further into the Diadasias and additional Euphorone species, please? So
we have Diadasia specimens.
Okay, we can go into Diadasia. As far as species level, maybe I'll put that on our to-do list as one of the first species level groups that are requested.
We want to, basically we want to go through all the genera because that's usually your flow, right? I've got to be what genus, then what species, and then we'll get into species, and it looks like we're, 3 or 4 more sessions.
Yeah.
It feels like a psychological sessions before we can go to the species level.
And then somebody does want to know how to build a straw bale home, in which case I'm going to stop recording and maybe we'll hang out while I do my post-class wrap-up thing. So thanks, everybody.
Did someone really want to know about straw bales?
Yep, it's in the chat.